Hello children. So I am once again back with the next video for the chapter life processes, and the main topic to be taken over here is the experiments to show conditions necessary for photosynthesis. In this video we will be discussing these key points. Experiment to show sunlight is necessary for photosynthesis. Then an experiment to show that chlorophyll is necessary for photosynthesis. Third, to show that carbon dioxide is necessary for photosynthesis. And we will be learning about the starch test, which will be common to all the three tests which we are going to discuss in this particular video. Now, we know that there are four conditions necessary for photosynthesis to take place, which include sunlight, chlorophyll, carbon dioxide and water. Here we will be discussing these three experiments. We do not have experiment for water because we cannot deprive the plant of water. It is always there in it. Hence we have these three experiments. We will begin with them one by one. Now, what happens during photosynthesis? We know that during photosynthesis, the plant manufactures glucose. We have done the equation for photosynthesis in the previous video. Link you can find in the description box. So, during photosynthesis, glucose is manufactured and it is transported to different areas. Glucose is a water-soluble form. Therefore, in order to be stored into the plant, the glucose is converted into starch. The glucose gets changed to starch. And this is the reason that we perform the starch test. It means that if in the plant starch is formed, photosynthesis has taken place, and if no starch is formed, then no photosynthesis has taken place. Therefore, we perform the starch test to check whether photosynthesis has occurred or not. Now, the plants are continuously manufacturing food. So, food is already inside the plant. How are we going to test then? So, for that, the first step of each of these three experiments will be, the first step will be to de-starch the plant. To de-starch the plant. Okay? To de-starch means to remove the starch from the plant. And how this is done? This is done by keeping the plant in dark room for 2 to 3 days. Keep the plant in dark room for 2 to 3 days. When the plant will be in dark room for 2 to 3 days, the photosynthesis will not take place. Whatever food will be there in the plant will be utilized. Now the plant which I am using for my experiment has no starch in it. So when I will perform the experiment by providing these conditions, then if starch is made, then photosynthesis has taken place, and if no starch has been made, then no photosynthesis has taken place. I hope this is clear. So we begin with the first experiment to show that sunlight is necessary for photosynthesis. First step, we take a potted plant, a plant put in the pot, growing in a pot, and keep it in the dark room for 2 to 3 days to de-starch the plant. After de-starching the plant, we take the potted plant and we select any one leaf which is nicely exposed to sunlight. Alright. So let me draw a plant with limited number of leaves for easy. Say this is my plant. Alright. So we select any one leaf and we cover a portion of the leaf, either with silver foil or with black chart paper, or we also get Ganong's photo screen. A Ganong's photo screen is available which cuts the supply of light. So here I am taking a silver foil and we take a small strip and we cover the upper surface and the lower surface with the silver foil or black chart paper or this Genong's photo screen, and we put clips on both the sides so that this does not move. This entire thing is now not getting any light. So it means in some portion of the leaf sunlight is provided, and in this portion of the leaf no sunlight is provided. We keep the plant in sunlight for 5 to 6 hours. In this much time enough starch will be produced. After keeping so my second step becomes cover with the silver foil. Alright, cover the leaf with the silver foil. Then the third step is leave it for 5 to 6 hours for photosynthesis to occur. After 5 to 6 hours, we will pluck this leaf, okay? We will remove this screen, and now we have to perform the starch test on this leaf to check whether starch has been formed or where starch has been formed and where not. Is it clear? Now we are coming to the steps of the starch test. These steps will be common to the other two experiments also. So after this has been done, now we will move on to the starch test, in which we pluck the leaf which we have taken for our experiment, and first step of the starch test is to boil the leaf in water. So now here we are starting the starch test, in which the first step is to boil the leaf in water. Every statement, every test has a reason, and if an activity based question is asked, these are the type of questions which will be asked from you. So first is boil the leaf in water, and why is it boiled in water? To kill the cells so that the cell membrane becomes permeable and allows our experimental things to go into the cells, so that when we are trying to change the color, it goes easily into it, or when we are trying to test. And secondly, by boiling the leaf in water, the cells are killed. As a result, now the translocation of food will not take place. Otherwise, if my food has moved from one place to the other, my experiment results will not be justified. So this is the first step. After this, the second step is boil this leaf. Boil this leaf in alcohol over water bath. There are two things. We have to boil this leaf in alcohol, and second thing is over the water bath. So again, why is it so? The leaf is boiled in alcohol to dissolve the chlorophyll. The chlorophyll comes out from the leaf when boiled in alcohol. It decolorizes the leaf. Why this decolorization is required? So that when we when we add iodine to change the color of the leaf, the color is nicely visible, because that green colored chlorophyll may act as an obstruction in the color change, or means the color change would not be visible clearly. So this is the reason for decolorizing the leaf. Second reasoning question which comes from here, why alcohol is boiled over water bath? Alcohol is boiled over water bath because alcohol is highly inflammable, and therefore if boiled directly, it may catch fire. The next step of the starch test is to wash the leaf in cold water. Then again Why the leaf has to be washed in cold water? Because when it is boiled in alcohol, the leaf becomes brittle. It becomes brittle means easily breakable. So to make the leaf soft again, it is washed in cold water. I hope all the reasonings are becoming clear. They are very important. And then finally, to the leaf we add few drops of iodine. Iodine has a property that it gives blue-black color with starch. So when to my decolorized leaf I will add starch, this is what I am going to observe. This was my leaf. This portion was covered with the screen. So on testing for starch, when we add iodine to the entire leaf, we will see these two portions will turn blue-black, but the middle portion where there was no sunlight will remain colorless. This is because this portion did not get sunlight and hence could not manufacture food, or no photosynthesis takes place without sunlight. This is my first experiment. I hope it is clear to you. Then we will move on to the second experiment to show that chlorophyll is necessary for photosynthesis. Now for this particular test, we need a plant in which the leaf has double color. You must have seen plants with double color. Very common example I will give you is the money plant. Money plant you all must have seen. In the money plant, some portions of the leaf are green and some are colorless, or you can say white. Bougainvillea also has variegated leaves. So some portions green, some portions white. Now when we have to perform this experiment to show that chlorophyll is necessary for photosynthesis, as I told you, so now we are discussing this one. As I told you, my first step will be to de-starch the plant by keeping the plant in dark room for 2 to 3 days, because we have to remove the starch. After the plant has been de-starched for 2 to 3 days, the pot is brought in sunlight and kept in sunlight for 5 to 6 hours so that photosynthesis can take place. Now say this is my potted plant and this is the leaf which I have selected, and I see this is a variegated plant I told you. So in the variegated plant, some portions of the leaf, say this outer margin is say white and the inner part of the leaf is say green in color. Same is with because the plant itself is variegated like this. I leave the plant in sunlight for 5 to 6 hours for photosynthesis to take place, and because the plant is already de-starched, after 5 to 6 hours I will perform this starch test on the leaf. First, boil the leaf in water. I have told you why. Then boil the leaf in alcohol over water bath and wash the leaf in cold water. Add few drops of iodine to it. In this particular experiment, before performing the starch test, one little thing we have to do. And what is that? We have to pluck the leaf, and on a sheet of paper, we have to draw the outline of this leaf, and we have to draw the outline of the green and white demarcation. This is for comparison. Why? Because now when we will decolorize the leaf, we will not have an idea where it was green and where it was white. So when we will get our starch test, we will compare it with this drawing. So finally, on adding iodine to the leaf, what will be my observation? The observation will be that the portion of the leaf which was green, the inner portion, will turn blue-black because it had chlorophyll, and the portion of the leaf which was white will remain colorless because it did not have any chlorophyll. So this experiment goes on to prove that chlorophyll is necessary for photosynthesis. And now the last experiment to show that carbon dioxide is necessary for photosynthesis. Again, my first step will be to keep the plant in a dark room for 2 to 3 days to de-starch the plant. Then we will bring the plant out in sunlight, and in this particular experiment we have to provide carbon dioxide to one part and we have to not allow carbon dioxide in the other part. So here I am going to use a chemical potassium hydroxide, chemical potassium hydroxide, because potassium hydroxide is an absorber of carbon dioxide. So I take a small flask, and in that flask I take a cork with one slit. I pass this leaf through that slit. See how? This is the flask, this is the slit, and here is the flask which we can keep on a stand. Alright, and in this flask there is potassium hydroxide. So now what is going to happen? The portion of the leaf which is inside the flask will not get carbon dioxide, while the portion of the leaf outside the plant is getting carbon dioxide. We leave this setup in sunlight for 5 to 6 hours for photosynthesis to take place. After that, we remove this setup, we pluck this leaf and we perform the starch test. Same steps as I have told you. On reaching the final step, the leaf is decolorized. On reaching the final step, that is the testing the leaf for photosynthesis. So suppose this was the place where it was attached to the plant, and this was the portion which was dipped in potassium hydroxide. So what is my observation going to be? The portion which was outside the flask will turn blue-black because it is getting carbon dioxide, and the portion which was inside the flask had no carbon dioxide, therefore will remain colorless, to show that carbon dioxide is necessary for photosynthesis. All the steps, all the reasonings I hope are clear to you. If you have not subscribed, please don't forget to subscribe to my channel so that I can help you in your journey through class 10th. Share it with your friends and don't forget to like the video. Thank you.